How's it going, everyone? Vincent here, and in this video we are going to be talking about the central dogma of biology. And if we go back to the 1950s and before the 1950s, scientists and biologists had this huge question on their mind, which is, how do your cells know how to make stuff? Because somehow one of your cells, like my skin cells right now, are falling off. Tens of thousands of my skin cells fall off every single second. So how are my skin cells able to replicate themselves and be able to produce all the things that they need to survive so that I can still have skin even though I get cut or I'm just shedding dead skin cells? And it was a very perplexing problem, and for the longest time people kept thinking that proteins themselves were the mechanism by which the information was stored to make more proteins. So proteins had the info to make more proteins. And this line of thinking was part of the reason why it took a long time for people to eventually realize that the actual source of information was nucleic acids and nucleotides like ATGC that you'll commonly hear, and that's regarding DNA. And so what Watson and Crick did is that they were proposing a structure for DNA, and it had to be this thing that was super compact and able to basically store these characters of information to code for things. And an excellent analogy to DNA and your genome as a whole is a book. And the reason it's such a great analogy is that, our alphabet has 26 characters. Your alphabet that spells your genome really has 4 characters. It's ATGC. So instead of, you can just replace everything with ATGC, and that's literally one character in this book. And then your DNA is considered this whole book. Your whole genome, the human genome, is about 3 billion base pairs long. So if you had a book with 3 billion characters, this is your genome. And so your genome, your DNA, lives inside of your nucleus. And these terms get a little bit scary, but basically the nucleus is nothing more than a little subregion inside of your cells. And in many ways you can think of it like a bank vault, because your cell is, this is literally, your genome is the most important thing from a biological perspective, because, and some biologists make this argument, the only purpose of life is to get genes, or your genome, to propagate through time. And the conservation of genes, this is the purpose of life from a strict biological definition. And there's even more analogies that we can draw to our genome in books, in that when we are looking through books, books have sentences, they have paragraphs, and so a paragraph, a bunch of letters, these nucleic acid letters, make genes. And these genes, they come together and they form chapters, and these chapters form books. And so these, there is a hierarchy of organization here, in that as a whole we have our genome. And so this genome is living inside of your DNA, or is living inside of your nucleus. And because it's so important that we protect it, because the last thing we want to have happen is our genes get mutated. Because when our genes get mutated, we no longer know how to make stuff, or we think we'll know how to make stuff, but we'll make it wrong, and this can lead to things like cancer. And so it's very, very important that the genome as a whole is preserved. And so what your cells do, and basically what Watson and Crick did, is that they were able to figure out that, how are these letters coming together? How are we forming these sentences? How are we structuring our DNA in the form of a double-stranded antiparallel helix? And so that is what Watson and Crick did in their Nature paper back in the 1950s. That was a pivotal moment in biology in terms of our understanding of how are we organizing genetic material using nucleic acids and not proteins, because people kept thinking it had to be proteins, when in reality it's actually just ATGC in some kind of sequence forming your genome, which is this book, and this lives inside of your nucleus. And so because this book is so important, we don't want to just hand this book off to someone and say, hey, go do with it what you want, because they might damage it, they might rewrite stuff, they might delete a page or two or a couple letters, and this would be catastrophic, because then we wouldn't know how to make things that we used to know how to make. So what happens inside of our cells as eukaryotes is that we have, outside of our nucleus, a thing called RNA. And so RNA is kind of like a cheaper version of DNA. This is like a printed text, very formal, very, stable, strong, gonna last a long time relatively. The RNA that comes out of your nucleus is stuff that's like kind of this handwritten scratch stuff that is very, it's not perfect. It doesn't have that great of a shelf life, but it gets the job done in terms of telling someone or something how to make something. Because at the end of the day, your genes are coding for proteins. And that's going to be the next step in this chapter. But basically, what we have here is we've got DNA that's super valuable, that is locked inside that bank vault. And then what we have is RNA that's not as valuable because it's only single-stranded, not double-stranded. And that single-stranded RNA is just going to leave your nucleus and then go outside into the cytoplasm, which is the area outside of the nucleus in the cell. You're still in the cell. And it's basically going to go tell a ribosome how to make something, because the ribosomes, their job is to make the proteins given the instructions to do it. So this would be the instruction set to make some kind of protein. And so in this example, I'm going to use a banana because I love bananas. But basically what would happen is the instructions for coding the protein, which I'll just represent as this banana, are going to be taken in by some ribosome. The ribosome is going to say thank you. It's going to look through all of the letters that are on this piece of paper, cheap single-stranded paper, and it's going to then translate that into the protein itself. It's going to make this actual banana at the end of the day. And so the key thing to make note of here is that the process as a whole took place is we have this book, which is sacred. It's locked inside of our nucleus. We don't want anything bad to happen to it. And inside of our nucleus, something happened in which we took out a section that we really cared about in the form of this note. And so on this note, we transcribed our DNA into RNA. RNA is the piece of paper, DNA is this book. And then from the nucleus, this RNA, this cheap thing that, it does the job of telling someone how to do something, left the nucleus, went into the cytosol, met up with the ribosome, and then it told the ribosome, hey, I've got something that you should translate into a protein. And so it translates the cheap note into protein. And so what you get out of that is stuff like keratin or insulin or any other kind of protein that you need in order to maintain homeostasis, to keep yourself alive and well. That's how your process works inside of your cells. And so this took us an extremely long time, but it is how it all works at the end of the day. I think it's beautiful stuff, and it is relevant even today during this COVID-19 pandemic, because we're seeing how basically what happens with this coronavirus, and I'll can do a follow-up on this too, but basically what's happening is the coronavirus comes into your cell, rewrites a bunch of this, has its own version of this piece of paper, hands this piece of paper to ribosomes, and then these ribosomes are able to make a bunch of coronavirus progeny. And so that is going to wrap things up for this video. I'll leave you on that cliffhanger. Thank you all for watching. Please stay safe, wash your hands, and I'll talk to you next time.